Thank you very much. Thanks, everybody, and thank you for taking time to step in and give us an opportunity to talk about our sequencing enrichment technologies. I'd like to give you an overview of how we use our technology for massively parallel single-plex PCR enrichment. And in particular, in today's talk, we'll focus on our sequencing-ready or seq-ready technology for targeted enrichment. So a little bit about our company before we get started, for those of you who are not as familiar with us. We've been around for a little over 10 years. We're actually based in Fremont, California. And in 2008, we launched our core technology, which was around the smart chip system. That was initially launched for real-time PCR diagnostics around expression analysis and genotyping. Later in 2003, under new management, we focused on the targeted enrichment area, applying our technology to that area. And then earlier this year, we made two additional major additions to our product portfolio. First of all, through the acquisition of the Apollo 324 system for library preparation through an acquisition from Intergenix Corporation. And secondly, and what we'll talk about today in greater detail, is the seq-ready technology, which we launched earlier this year, which is focused not only on custom panels, but I also talk about our fixed content panel for the BRCA1 and BRCA2 genes. This development and acquisition of technology over the last few years has resulted in a series of a product portfolio which spans many areas of the NGS enrichment and library preparation region. So, for example, in both discovery and in clinical applications, we have our Apollo system, which can perform library preparation on whole genome DNA, RNA-seq, ChIP-seq experiments, and also through sample prep from in RNA and RNA ribodepletion kits. Secondly, through our smart chip technology, we're focusing on target enrichment, and this is mainly focused on a clinical or CLIA-type environment for applications on targeted enrichment. And lastly, through our earlier technologies, such as the smart chip qPCR system, the same technology can be applied for both library QC, high-throughput genotyping, and high-throughput expression analysis. In today's talk, I'm going to focus primarily on the target enrichment portion of our product portfolio and its application to next-gen sequencing target enrichment. So why focus on targeted sequencing? Well, it's the case that next-gen sequencing is making rapid inroads into clinical testing. And while exome and whole genome garner many of the headlines in terms of their introduction into clinical next-gen sequencing, a majority of the panels and the next-gen sequencing which will occur in the clinic is currently and will continue to be around targeted panels. The reason for this is a numberfold. One is the complexity of data analysis around whole exome and whole genome analysis, data analysis and privacy concerns around analysis on such large data sets. And lastly, but not leastly, exome still can be challenged in terms of coverage and coverage uniformity over such a large region as the whole exome. So targeted panels will continue to play a major role in sequencing in the clinic for the foreseeable future.